Recording in progress.
Okay. Good afternoon, everyone. Hi. So today, after class, there's going to be no time for like student hours for questions and stuff. I have a meeting I have to go to right away, but remember this Thursday, we're going to have a Q&A session. The whole session is for you to ask questions, right? If you have something that's urgent and cannot wait, you can send an email. We're going to be using Clicker today, so let's get started. And then at the end, if I have time, I'll mention more about the deep question evaluation stage, which is going on right now. You have until Friday to finish that. Okay, so yeah, that's what I just mentioned. Q&A session on Thursday. So come with your questions. Come prepared with questions. I won't be able to stay after class today. Now, this week, your materials focused on mitosis and meiosis. I'm sure you all know both of those very well. And I think the materials this week went into more detail than you may be accustomed to. It was deeper than just like a general review. And actually, I hope it started getting you thinking about it. So today's activity is going to focus on these two processes and how they differ and what is the significance of the difference. So learning outcomes for this week, describe using diagrams. So diagrams and appropriate terminology, the sequence of cell cycle events involving DNA and chromosome replication through chromosome segregation. So diagrams, terminology, you're going to want to be able to draw these out, understand them enough that you could de novo draw them out, know the differences, and also use appropriate terminology when describing them. So compare and contrast mitosis and meiosis, and in terms of the process and also the outcome. Compare and contrast the timing and products of meiosis in spermatogenesis and oogenesis. Relate independent assortment and segregation to the behavior of chromosomes and the distribution of alleles during meiosis. Explain how independent assortment of alleles during meiosis can lead to new combinations of unlinked genes. Explain how homologous recombination can lead to the new combinations of alleles on the same chromosome, and explain how specific combinations of linked alleles in SNPs, which are a haplotype, right? So linked alleles and SNPs segregating often together constitute a haplotype and persist through many generations. So in other words, how they can be, then inheritance can occur again and again. Why does that happen? Recognize evidence of linkage in the outcome of crosses. Differentiate between the terms independently assorting genes and linked genes, and use and recognize conventions for representing linked genes. Be able to differentiate between the genotype, phenotype, clearly explain the relationship between genes and proteins. Well, sounds pretty straightforward. Okay, so meiosis is reductive cell division that produces gametes, gametes being the egg and the sperm, right? And so a human somatic cell, the cells that are generally your body is comprised of, they're diploid, and they contain 46 chromosomes, right? 46 chromosomes. We went over the chromosomes. We saw how 46, but there's really 23 pairs, right? Well, the sperm and the egg, as you know, they only have haploid set. What does a haploid set mean? What is haploid?
Hey, Professor.
Yeah.
Could you share your screen, please? On Zoom.
Okay. Yeah. Only one copy of each of the chromosomes. Yeah. Right. So they have only one copy. So they have half, right? They're haploid. They've got 23 chromosomes total, 23 chromosomes. Okay, so the sperm and the egg are haploid. The adult human, diploid. Okay, so what happens here? So how do you go from a diploid to a haploid to a diploid? You have your haploid sperm, as you know, fertilization, and essentially those 20, that haploid set of chromosomes from the sperm combines with the haploid set of chromosomes from the egg, producing a diploid
Bro, this is
set.
the second time my professor
So at
started
that
a
point,
Zoom
every chromosome, there are two copies. It's diploid. There's two of them.
without sharing his screen.
During fertilization, the chromosomes, the egg and the sperm unite
And
to, I don't know,
he records it on
chromosomes
Zoom,
unite,
but he like teaches
they
in person,
co-localize
So-
into the same,
right? So he can't hear
Nucleus.
any of us on the Zoom.
and hence starts the life of the diploid organism, right? What is that first cell called? The first diploid cell that's formed? The zygote. Zygote. And then the zygote goes on to develop eventually an adult organism. And so how does it do that? The zygote is diploid. And essentially after that, nothing changes, right? You have that diploid cell, and through many mitotic divisions, okay, you get an adult. About 50 to 70 trillion cells. Wow, think about that. That DNA gets copied many times to produce 50 to 70 trillion cells. That's kind of like the homeostasis. That's the adult is going to maintain that many cells. Some of them die off, some of them get replaced, so on and so forth. But anyways, it's through the somatic,
This guy's a bot, bro.
mitotic divisions that those somatic cells, they have produced all those cells.
Crazy, bro.
Okay, it doesn't happen in one day, it happens over a long, long time. And currently, like in your body right now, there are millions of cells turning over very, very frequently. Okay, so this growth that we see here is mitosis, the process by which a cell replicates its chromosomes is mitosis, then segregates them into two identical nuclei. That's mitosis. In preparation for division, what's division called? What is the term that the videos use for division? Yeah, cytokinesis. Great. The pinching off, you saw those little balls, they're kind of like molecular vibrations, and then they pinch off, and they're two cells. So cytokinesis. Great. All right. Let's get started here with the. Okay, after fertilization, the zygote divides mitotically. What does this mean regarding the cells of the zygote? That they have different genetic information because the zygote will form specialized cells? Or is it that they have the same genetic information? 5, 4, 3, 2. Right. 86% of the class says, yeah, it's B. They have the same genetic information. So that's mitosis is going to generate identical chromosomes. We know that, right? We know that. So mitosis creates daughter cells, and they have the same genetic information as the parent cell. All right. So on your own, go ahead and do this. So genes A and B are on the same chromosome, while gene E is on a different chromosome. Niru is heterozygous for all 3 genes.
A flashlight still doesn't work?
Draw out what these chromosomes and their respective gene locations would look like after the cell has passed through S phase, but not undergone cell division. So let's just say, draw them how they look like, I don't know, like metaphase or prophase. Okay, take a few minutes. I'll give you 2 minutes. Draw it out.
Excuse me, Professor?
Later in the course, things get much more complicated. You're going to want to do these exercises to really get comfortable with this. You may think you know it, but then when you try to draw it out, you may be like, more complicated. All right, so you got it drawn out, right? Yeah, just a few genes, just 2 chromosomes, and okay. Now, what I want you to do is, I want you to share and compare. So in groups of 2 to 3, I want you to compare your drawings of the chromosomes, focusing on genes A and B. Genes A and B. And which are on the same chromosome, right? So this is a heterozygous individual. You're supposed to draw 2 alleles. Did you draw it the same way as your partner or as the other people in your group? Do you have alleles A1 and B1 on the same homologue? I think everyone's confused. Did I lose you? What happened there? What happened? Let me know. Let me know. What happened? Okay, I see some discussion going on.
Excuse me, Professor.
Did you draw them the same way?
Bro, my professor's actually autistic.
Again, if you have an open seat beside you, you know, just wave someone over. This is really, it's a lot more helpful when you do discuss your results with another person. You learn from your group members. All right. So,
Oh my God.
does anybody want to, you know, let me know? Did you draw them the same way as your partner? Yes?
Oh my God. That hurt. That hurt.
No?
Oh
Okay.
my God.
So the 80% of the class that didn't raise your hand is somewhere between yes and no. So I'm just very curious about that. Can I
What the fuck is this
maybe
guy doing?
understand a little bit about what's going on there? No, don't ask. Okay, all right. Okay. Yeah, so I mean, yeah, maybe you did. We only got there's only what 3 genes here, and we're just talking about the 2 of them. Like if we look at A and B, did you put them on this A1 B1? So the neuro's heterozygous. So you got 2 copies of A, right? You're gonna have A1, and then you have A2. Those are your 2 different copies. And then you have B1 and B2. I guess what the question is did you put A1
Jeez.
and B1 on the same chromosome?
See, that's what I gotta do.
Yeah, a lot of guys.
I gotta-
That's the way I would
I
have
gotta
done
put
it.
TTV.
Why not? Did anybody put A1 with B2? Yeah, why not? Sure.
Wait, they
There's
actually
nothing wrong.
redid
It
everyone?
didn't say you couldn't. So, yeah,
What the
people made it
fuck?
drawn it different ways.
What does
Yeah.
that even do?
Excellent. Okay, now look at this. Neuro's genotype is A1B1. What's that? Slash A2B2. And then semicolon E1 and E2. Okay, so what does writing the genotype this way tell you? Let me pause for a second there. If you were to draw them based on this, could you and your partner have different drawings? No. Okay, so let's unpack that. What's going on here? So what does the writing the genotype this way tell you? Anyone? We all watched the section about genotype annotation. Yeah. Let's start small. Like, I guess just, how about this? You just tell me, what is this first part here? A1B1 dash A2B2.
I
What
think I'm
is
good at
that
it.
telling us?
Yeah, even if I'm not streaming, they can just check out my channel.
Did you say A1 and A1?
But yo, what's dumb about PlayStation is that every time you switch games, you cancel the stream. Yeah.
Same chromosome number, like chromosome
Yeah,
1.
so if I
yeah.
want to like swap games, I would have
Yeah,
to
sweet.
buy
Yeah,
a
excellent,
capture
excellent,
card.
excellent. Did everyone get that? So let me repeat if I may. So A1 written beside B1, that means they're on the same
Yeah,
homologue.
yeah.
Let me see if I can write stuff here. Is it going to cooperate today or not?
No, but you can always just swap the games after.
Pointer
Like,
options.
on
See, I
Twitch.
can't get a pen. I can't get this.
Yeah, like
I think
manually
it's
switch it.
because of Zoom. So let me try and do it through Zoom.
You don't know what a capture card, Bobby, is?
Probably am going to want to draw at some point.
No. Yeah, like Elgato that records like your screen.
Zoom, what are you doing?
Elgato is, but you can get ones for like $50. But it's
Wow.
still like 50.
Zoom, are you there? Oh, that's why. Oh, okay. Okay, so apparently through Zoom, if I can find the whiteboard.
Oh my
I
god,
always
wait.
forget where this
Wait.
pen
Does
thing
my
is.
professor know that he's not share
No,
playing?
it's not working. So pointer options. Oh, I still can't draw, huh? Okay. I thought I could do it through Zoom. I thought last time I did it through Zoom somehow. Didn't I do that?
Wait, what he's talking about? He's doing it through Zoom. Does he not know?
Okay, before I lose your attention too much, I might as well get back to it. So, thank you very much for
No.
the answer. The answer, I'm just gonna draw on the
This bitch
board here.
boy.
Sorry for
Alright, what
those
were you
on
saying?
Zoom. That's like, you gotta come to class. Oh,
Wait.
and there's no, you got one? Ah, thanks. Saving me here. Yeah, sweet.
Wait.
Oh, no, this is a permanent
no.
one. No?
Donate.
Let's, everybody go like this, just in case.
I'm gonna be like first goal to get a capture card.
Okay. Hey, all right, we're in business. Thanks. You're on the team. I'll have them send you the check in the mail. Okay. So A1, B1 are on this, they're the same homolog, right? That's what that means. So I got my A1, B1. This
No, but then
is
they would have
meaning
to
it's the same
be able to
homolog.
stream the entire PS5.
I probably wrote it in it. I'm sorry here.
Maybe.
Okay. The same homolog, right? And then A2, B2 is another
yeah.
homolog, right? So this is A2, B2. And so look at, this is homolog 1, homolog 2. Okay, so look at how they wrote it here. So what is this? This, like, I'm calling it a slash. What is that slash telling? Like, what does that mean? That separates these two? Yeah. Yeah, very good. So it's telling us those two on each side of this slash here, those are homologous chromosomes. And it's telling you on one of those stretches of DNA, which constitutes a homolog, we have these two alleles. And on the other homolog, which is a separate molecule, we have these two alleles. All right, great. Now, what about this? What does this semicolon mean here? Yeah. That's telling us this gene E here, it's on a different chromosome. So this might be chromosome, I don't know, 7. And this one here might be chromosome 13, for example, right? Okay, very good. Okay, so, and I think I wrote that down here. I didn't even have to draw it out, but I didn't. So this is a homolog. This is the other homolog. And this is a different chromosome. Okay, so does this tell us additional information that I didn't tell you previously? Yeah, what did it tell you that we didn't know before? Yes. Yeah, allele 1, A1, that allele is on the same chromosome as B1. So contrary to before, how people were drawing A1 on the chromosome with B2, and some people were drawing A1 with B1, here the annotation specifically tells you additional information, and it's important information. Why do you think it's important? What is the connection between A1 and B1? Okay, let's move on. I'll give him a chance to think about it a little bit here first. Okay. Okay, so Nero's genotype was this. And then we have, is this how you drew it? You have your one homologous chromosome here, another homolog. Then you got your, sister chromatids. And an A1, B1 on the same piece of DNA, A2, B2 on the other. Okay? So your chromosome that you originally draw, it could look different because we didn't initially say it in this particular terminology, right? So there's other ways that this could have been drawn if I didn't specify what chromosomes they're on. Okay, so now let's look at this. Genes A and B are close together on the same chromosome. The genotype of two individuals, both of whom are heterozygous for genes A and B, is given below. Same genes, different person, different genotype. Is Sveta just as likely to produce gametes with A1, B1 as Jackie? Yes or no? Okay, 5, 4, 3, what's going on there? 2, 1. All right. This is wow. This is we got 54% saying yeah, and then we have 45% saying no. All right. Okay. Does anyone want to delve into this a little bit? I always like I think this style of this course is kind of leave you hanging until the end, but I think a lot of times people get lost. So I'm going to pick and choose. Sometimes I'll leave you hanging, and then we'll get to it at the end of class. Sometimes we're going to get straight to it. So yes. Hold on a second. Guys, hey, everyone in the back and stuff, please respect the other people in the class. Yes.
Huh?
Okay.
I am recording it. It's on. I post it on my YouTube.
Yeah.
How? Yeah.
Yep.
Nobody's gonna know.
You're-
Who the fuck
you're
is
right
gonna
on.
find my YouTube? Yeah, who the fuck is gonna find my YouTube, man?
Very good. Very good. I know everyone couldn't possibly hear that because we're not mic'd. We should have one of those mic cubes we throw around whenever. That would be awesome. The university should do. I actually think I might try to push for that. That'd be great. Yeah. So what's your name? Christian. What Christian was saying was that like A1. So our question is about A1 and B1, specifically this allele combination, right? And so look at Sveta. A1, B1 already has it. Jackie, on the other hand, A1, B2, so needs to form it. It's not a big deal, need to form it. That can happen. But the genes are close together on the chromosome. What does it mean when genes are close together on chromosomes. What happens to those genes when those alleles of the alleles, what happens to those alleles when they segregate? Yeah. Okay, so yeah, dependent. Yeah, I see what yeah, the dependency of the. Okay, so if they're close together, chances are, well, they're linked, right? They're linked, and they'll likely show a linkage effect. And what is a linkage effect going to be? Yes. Okay. Recombination, crossing over. If they're close together to one another, is there, okay, and that's recombination, you're right. That's what Jackie needs, right? Jackie needs to have recombination in order to get A1 and B1 on the same chromosome. You follow me? Yeah. Okay. Sveta doesn't need anything. They're already there. Jackie needs cross recombination to, and so you can picture this. You know, we have our draw it out here, like this. That's my. These are my chromosomes. These are chromosomes, guys.
Wait, where's the gender
Here.
progression?
And
I see.
so A1 and B1. I would be the other A1, B2, A1, B2. That's what she had, right? A1, B2, right. And then A2, B1, A2, B1. We're talking about Jackie here.
Why are you
Okay.
trolling?
And so in order to get
You killed yourself,
A1 on the same chromosome as B1,
Jeez.
we'd have to have a recombination event, right? You follow me? Good. Okay. I'm not going to get too much more into it than that. We're going to develop this as we go. Okay. So, are they as equally as likely to have the gametes, to produce gametes
Oh, you're
Maybe
running off the right shit.
as A1 with a A1, B2 combination, the A1, B1 haplotype?
Jeez.
So you half the class is saying yes, half the class is saying no.
Jeez.
After drawing it out like this, does it make it any more clear what the correct answer is?
He's gonna tunnel
There
your
is
ass,
only
bro.
one
He's
answer.
gonna tunnel your ass.
If they're close together on the genome, is it going to be likely that
Yeah,
recombination
I know.
happens to put this allele on the same chromosome as this allele, or is it going to be not so likely? I'll give you another scenario. I have a dartboard that's this small, okay? And in order to get recombination, to hit the bullseye, I got to get it in the dartboard, right? Am I more likely to get recombination, to hit the bullseye? Am I more likely for it to happen if the dartboard is small like this, or if the dartboard is huge like this? In other words, if the genes are further apart versus if they're very close together. Further apart, much more likely to have recombination, which is what Jackie needs. These ones are close together.
Where the hell is Ali?
And we'll come back to it. I think it's not hitting yet. Okay,
I'm trying to play green.
let's revisit Niru. Genotype A1, B1, homolog. A2, B2, E1, E2. Use your earlier diagram of the chromosomes as a starting point and determine the movement of chromosomes. So with the respective genes, alleles, okay, so through meiosis, go through meiosis with these. So I would, start close to the beginning and try to work your way down to the reductive cell divisions that happen to form the gametes. Okay, in groups of 2 to 3. So take that original chromosome that you had drawn And now work your way down to making those gametes. You can have meiosis, just very generally. You don't need to go through every phase. What's going to happen after meiosis 1 is done? And what's going to happen after meiosis 2 is complete? What are those chromosomes? And as you go through, I want you to also, where was I here? Consider these questions. So, what phases are important for how the chromosomes or those alleles sort into daughter cells? So as you're going along, I want you to pay attention to what phases are important. And also, in which phases do the following appear? These should be checkpoints of things that you would be drawing into your schematic, into your figures. So recombination, independent assortment, sister chromatid segregation. Where are those going to take place? So draw at least enough images, at least enough steps where you're incorporating those. Okay, go. Work together in groups. It's not easy. This is not an easy task. Okay, just a few minutes.
Wait, this horse is Maurice? Was he always there? Why did they name him Maurice?
Okay. All right.
Oh my god. Bro, this guy's getting shit on.
Okay, considering genes A and E only, which of the following processes would generate different combinations of alleles of these two genes? Just A and E. They're on different chromosomes. Okay, 5, 4, 3, 2. Oh, sorry, I clicked it. I'm sorry, guys. Okay, so most people, 48% are saying D, so there's 2 of the above. 17% are saying sister chromatid segregation. 29% are saying independent assortment. 14% are saying recombination. All right, so considering genes A and E only, if we're considering genes A and E, which of the following would generate different combinations of alleles of the two genes?
Yeah, how do you not see it?
What is independent assortment? Yes.
Why didn't you take the hit?
Okay. Independent assortment. Okay, whenever the chromosomes line up in metaphase and they're ultimately going to be pulled to separate poles of the cell, so the cell can pinch off and make two new cells eventually. It's the independent assortment is that the chromosome, one homolog of chromosome 1, for example, it will segregate. It'll go to one particular pole independently of all the other chromosomes. It doesn't matter what those chromosomes are doing. In other words, whether the chromosome number 7 from mother is coming along with it, or chromosome 7 from the other parent is coming with it. That doesn't. There's no influence between the chromosomes. That's independent assortment. Okay, so you have two copies of any particular chromosome, right? Like, let's just say, this is chromosome 3, 2 copies. A1 and B1 are going to get pulled to one pole, and either E1 or E2 are going to get, that's independent assortment. So either of these could go to that pole. What about recombination? What's recombination? Yeah. Crossing over. What's crossing over? Yeah. I like how you say two chromosomes. You didn't specifically say chromatids, but two chromosomes exchange genetic information. Okay, and in recombination, they recombine. They can transfer genetic information. So now all of a sudden it can make two combinations of alleles. Okay, is that going to affect what combinations of A are going to go with what combinations of E? Okay. All right, I'm just going to, oh, my drawing thing, the drawing thing is not working here, guys. It's locked me out for some reason. It's not showing up as an option. So I don't, so I'm going to cheat. Not cheat. I'm going to use some drawings from previous. Here we go. It's not because I'm not that good at drawer and I can't figure out how to use the software. I'm just going to pull this up. How do the chromosomes align in mitosis versus meiosis? Because, okay, let's just back up for a second here. Whenever we talk about independent assortment, are we talking about mitosis? Okay. I got a small fraction of the class actually no. Okay. Mitosis, remember what we said. Remember what we said in mitosis is that one parent cell produces two identical daughter cells. If you had things assorting independently, would the daughter cells be identical? Okay. All right. Meiosis, on the other. Meiosis, do we get independent assortment during meiosis? Is it of significance in meiosis? Okay, I'm getting some yes. Okay, thanks. Thank you. Good. So now let's look at the difference between the way things line up on this metaphase plate, the chromosomes, the way the chromosomes line up, right? So in mitosis, what's going to happen is you have a chromosome 3 that came from the egg, and it looks like this. That's one chromosome. Remember, it's attached at the center. And then you get a chromosome 3 that's from the sperm. Those are the same chromosomes, right? But they align like this. In mitosis, they align. How do they align? Head to tail. Head to tail, head to tail, head to tail, head to tail. And then what happens whenever they get pulled apart? They separate and they go to opposite poles. What went to the opposite poles is identical. The chromatid from the sperm's chromosome 3 one chromatid went to one side, one chromatid went to the other side. And the same thing for the chromosome 3 that came from the egg. Split in half. One went to one pole, the other to the other pole. And those poles ultimately make new cells, right? So they're identical. But what happens differently in meiosis? Yes. Okay. That's very cool. The chromosome, the homologous chromosomes, they get paired up. They don't go head to tail. They do something like this. Okay. They pair up. So remember the chromosome that came from the egg? It pairs up with the one that came from the sperm. Side by side, not head to tail, side by side. Here's your plate right in the middle. Sorry, I didn't write mitosis in there, but meiosis, right? This is meiosis. And then the same thing happens for your chromosome 7, which is shown here. Now, what's the significance of this? On one side of the line, chromosomes are going to go to one pole and end up in one cell. On the other side of the line, they're going to go to the other pole, right? And who's to say that the E1 chromosome is going to go to the same pole as the A1 chromosome? Right? The E1 could just as easily go to the same pole as the A2. It does, though they may align differently. We don't know. We don't know on what side of the plate they're going to end up. Okay, so this is independent assortment. How this one segregates the alleles from this particular chromosome, it's independent to how these segregate. And that creates combinations, right? Only happens in meiosis. Doesn't happen in mitosis. It creates genetic variation. Why does it create genetic variation? I'm not talking about creating new alleles. What am I talking about here? What are some terms that I mentioned earlier that it creates? Yes. Different combinations of alleles of the genes. Haplotypes. Maybe not. Maybe. Haplotypes would be left linked. But anyways, different combinations of alleles ending up in the cell. Different from the parent cell. Okay? Never mind the haplotype thing, we'll come back to that. Okay, so now you know the difference, and this is how you would draw them differently. Mitosis, you draw something like this, and the E chromosomes would also be head to tail, whereas here, in meiosis, they're side by side. On your own, is Niru just as likely to make a gamete with A1 and E1? Just on your own, try to do it on your own. As she is to make a gamete with A1 and E2. Five, 4, 3, 2, 1. Yeah, as soon as I started counting, people panicked and they started changing their answers. We had upwards of 92% of the class that had the right answer. Pumped up, love it. We're down to 85 now. But anyways, I won't look at that. 92%, awesome guys. Okay, is Niru likely to make a gamete with just as likely to make a gamete A1E1 as she is to make a gamete with A1E2. Why did the most of the class say, yeah, it's equally as likely? Why? All right, let's play some music, back it up. All right, let's do this. Why? Why, guys? Yeah. Yeah, they're on different chromosomes, right? And remember the alignment of the plate. It all comes down to that alignment of the plate. And so you had your, bring it back here for a second. You got A1 on one side, and we're asking you, essentially this question is asking, well, is it equally likely that it could end up with E1 or E2? If you go back and look, we said, like, yeah, we don't know. It's equally likely that E1's going to end up on one side of the plate as it is to end up on the other side, right? Boom, got it. Okay, that's great. So that's what I got really psyched up about that. That's perfect. I love it when I see coherent, like a consistent answer among most of the class. That's great. Okay. Now, is Nero just as likely to make a gamete with A1 as E1 as she is to make A1 to G? Why am I not? Would your answer be the same if I asked you, is a gamete with B1 and E1 just as likely as a gamete with B1 and E2? Yes, let's do, yes, it's equally as likely. I'm talking about B now, and again with the E's, they're on different chromosomes, not fooling you guys. You already, yeah, there's no difference there. Okay. Now let's consider all three genes. What processes allow Niru to produce gametes with different allele combinations? So gametes like this. What processes? Okay, just very, very briefly, go. Oh. Back up. Go. 5, 4, 3, 2, no, don't change your answer. No. All right, we had up at 75% of the class was, that's okay, it's okay. You know, I just want you to participate, but I really like it whenever you guys are, you know what's going on. So we're down to 67%, say C, okay? 67% say E. So why would we expect both recombination and independent assortment to produce these alleles with these combinations? Remember Niru's starting genotype. This is what's present in the somatic cells, all the cells of Niru's body, including those germline cells that are going to produce gametes. Okay? So why yeah. Mm-hmm. Fantastic. You went right to it. So hold on. A1 and B2, A1 and B, there originally was A1 and B1 on one chromosome, A2, B2. If you see this notation here, it means that A1 and B2 ended up on the same chromosome, right? Okay, in order for that to happen, you would have to have recombination between these two chromosomes here. Okay, so that was recombination, and then the independent assortment. How do we get the different, yes? Yes. Yeah. Very good. So if you just look at the A1 and the E's, you got one of them has A1 with E2, one has A1 with E1. That's independent assortment, right? That's how you could get those two different combinations because they're different chromosomes. Very good. Okay. Okay, if recombination did not occur, could Nero produce gametes with the genotype?
Yeah, Dama.
A1,
Greenhill.
B2, E1. Oh, I gotta click the thing here. Yeah. Okay, go. Recombination did not occur. Okay, good. 5, 4, 3, 2, 1. Every time I count, the right answer goes down. Don't panic. 74% say B. B. If recombination did not occur, could neuro produce gametes with the genotype A1B2? The answer, no. You guys are saying no. Why are you guys saying no? Well, we're talking about gametes. Gametes only going to have what? A gamete is going to have actually only one of these chromatids, right? It's only going to have one of the chromatids. And so if recombination did not occur, how could you get A1 and B2 together? So in other words, how could one of these bars here that start with A1, B1, how could it become A1, B2? It's not possible, right? There would have to be recombination between the chromosomes in order to exchange. Okay, so this is a recombinant, or it's called a recombinant. It's also called a non-parental haplotype. They're alleles, combinations of two different genes, and they're on the same chromosome. Okay, so non-parental combination of alleles is another way you can think about it. And so what had to happen there? So on your own, consider the chromosome pair with genes A and B. If recombination occurred between A and B, what would the chromosomes look like after? What would the chromosomes look like? Don't pay attention to that purple squiggly drawn on there. Can't for the life of me figure out how come it's blocking me from drawing it. This is, I'll have to figure that. I'm gonna have to have somebody show me how to do that. Okay, what happened? Okay, so did you draw it out? Okay, now what recombines? What recombines? How does this look? What happens if there's recombination here between sister chromatids? Listen to the language and look, I'm pointing between sister chromatids. We get recombination. Anything significant happen if there was recombination there? No, it doesn't matter. You're taking two things that are identical and switching them. They're identical after, right? Okay, because this DNA, you got to remember, resulted during replication, and these were semi-conservative. So these are copies of the initial parent DNA. They're identical, okay? And over here, separately, these are identical to one another. There's no difference. If they cross it back and forth, doesn't matter. That synaptonemal complex forms between these chromosomes, between these homologous chromosomes, aligned up at the plate, side by side, and then you have proteins forming, making the synaptonemal, and then there's going to be exchange of genetic information between the chromosomes. Okay, so this X that's drawn here, I don't know, that doesn't really make much sense. It's going to be an X that goes from here, and somewhere in between the blue bar and the red bar, we have a recombination event. Okay, and let's see if it, yeah, I apologize for the whole pen thing. I'll figure it out for next class. I promise. But the recombination would ultimately occur like this. This whole chromosome, imagine there's only one recombination event, and I'm drawing along the length of the DNA. And what happens is I hit this blue A1 allele, and in between that blue A1 and the pink, the B gene, I'm gonna recombine. So this chromosome, the proteins, the synapple-nemal complex, are gonna exchange. They're gonna detach one chromosome and connect it to the other, and vice versa. Okay, so now if I was following along the length here of this chromosome, I would be switching over here, and I have the pink on the same DNA as this light blue. I would have also the red on the same DNA over here. That was exchange of genetic information between non-sister chromatids, between
5 gigs should be quick.
homologous chromosomes. Okay, that's how you get that. All right. So consider the chromosome pair with genes A and B. If recombination occurred between A and B, what would the chromosomes look like afterwards? Okay, so this is what they would look like afterwards. This is exactly what I was trying to draw out, but I'm unable to draw this thing. That you got recombination happening. So now the light blue is on the same chromosome as the pink. You see how the pink got swapped over here? We got our pink allele here with our light blue. And then we got our blue allele here with our red. Now, you got to think back to the parents. Think back to the parent cell. The parent cell, it had this one combination of alleles on one chromosome and another combination on the other chromosome. Those were both parental combinations. But now, through recombination, we have the formation of non-parentals or recombinants. And it's not everything that got recombined. Now, we didn't switch over, just the non-sister chromatids that were side by side had recombined. These two here, they remain parental. Okay, and remember, each of those is going to get pulled into a separate gamete. Okay, so you'll have parental gametes and you'll have non-parental gametes. Okay, so remember that recombination exchanges genetic information between two non-sister chromatids, okay? Look at the new combination formed here. We had A1B1, and now all of a sudden we have A1B2 and A2B1. Okay, so in groups discuss recombination has occurred between genes A and B. What do the daughter cells produce? So recombination has occurred, just like we talked about. Okay, what do the chromosomes look like at the end of meiosis 1? And what do the chromosomes look like at the end of meiosis 2? Go ahead, draw it out. Work together, it's not easy. Okay. Okay, so here's something for you to practice, okay? So you have a diploid cell, 3 different types of chromosomes. Draw how chromosomes align during metaphase of mitosis. We did that, right? We know, let me see if I'm gonna work around it's blocking me, but let's see if I do this. I think this is the way. This is the only way. So insert and Wow.
Huh?
I don't have the opportunity to. Oh, that might be why. I wonder if that's. You know what? Let's see if it works now.
Like, yeah, that's what I was looking at.
Ding. Ah, oh my goodness. It's because it
Yeah.
was locked and not editable. Okay.
I think store.
Uh,
Yeah, so survival is literally just
okay, so
just surviving.
draw your chromosomes and how they align
we
during
wanna start
metaphase
here.
of mitosis. You know what? I'm just gonna
And then we do
discard
skills.
this. I'm just gonna go back. Let's pop that up for a second.
Button to
Go
draw.
back
We learn our own.
to, where was it? Where did I draw that out?
Chill. You can do it. Teach me after.
Okay, this was mitosis, right? You have them aligned, the homologous chromosomes head to tail. And on meiosis, you have them side by side. The homologous chromosomes are side by side. Okay, at the end
a tutorial
of
contains a big
meiosis
portion of
1,
story mode.
what do these chromosomes look like? Have their homologs been pulled apart? The chromatids? The homologs have been pulled
If you
apart.
wanna
The
do
homologous
story.
chromosomes go to separate ends, right? So what your picture there, yeah, and okay,
All right,
right.
let me watch you and then you.
Okay,
host.
so your picture here. Oh no, I still haven't solved the problem. I don't know. Okay, going to the board. So your picture here, meiosis 1. I'm just going to put M1 for now because I don't have a lot of time. And so here's your plate. And your chromosomes, we said they aligned on the chromosome. These are homologous chromosomes. And so what are they going to look like at the end? Let's say they have A1 on this side, A1, and this is A2, A2, and the B's are B1, B1, B2, B2. So let's say that these have recombined. This is B2, this is B1. They get pulled. So at the end of meiosis 1, what is it going to look like? Essentially, you're going to have these chromosomes being pulled apart. So one's in one pole. This is pole 1. And one's in the other pole. This is pole 2. Okay, and so we still have those same combinations, right? A2, B2, A2, B1. Crossing over occurred, okay? B2, A1, A1, B1, okay? These are now separate cells, right? And now what happens after, that's a meiosis 1. What happens in meiosis 2? These sister chromatids get pulled apart. So essentially, your plate forms like this. And these guys form 4 new separate cells, right? This half goes into one cell. This one goes into the other. This one goes into one cell. This one goes into the other. Okay, so your chromatids get pulled apart at the end of meiosis 2. All right, back to neuro. For genes A and B, Neuro's genotype is A1B1, A2B2. Is one of Neuro's myocytes, that's the cells that undergoes meiosis, in one of Neuro's myocytes, recombination occurs between genes A and B. What proportion of the 4 cells produced from meiosis are recombinants? Five, 4, 3, 2, 1. Wow, 40% of the class is saying it's 1/4. 40% of the class is saying it's half. I just drew it out, and I guess my drawings are not very good, right?
Yeah.
Okay, so oh, it's brand new, is it? Okay, so this is what it would have ended up at the end of meiosis 2, right? So A1, B1 together, and then we have the other chromatid, which was A1, B2. We have A2, B1, and we have A2, B2. Which one of these are recombinant? Which ones are non-parental? What did we start with? We started with A1, B1 being on the same DNA, and A2 and B2 being on the same DNA. Which ones are new combinations? The two in the middle. What is that? Is that half or is that 1/4? It's half. Thank you. Yay. All right. So, meiosis 1, this is essentially what happens. At the end of meiosis 1, you get your chromosomes that look like this. And I'm going to post these slides so you guys have it. So no panic. At the end of mitosis 2, you see how we had the recombination here, and we're going to have those chromatids get pulled apart. So at the end, one chromatid gets pulled apart like this, other one gets pulled apart like that, and we have 50% are recombinant and 50% are not. Okay, hold on, guys. Let's slow down. We got 6 more minutes. Okay. Okay, so go. If genes A and B are immediately next to each other on the chromosome, how many of the whatever would you expect to be recombinants if they're right next to each other? 5, 4, 3, 2, 1. Okay. 50% of the class say that it's less than 1/2. That is correct. Only 50% of the class said that. Why does it matter? I just showed you in this example, it was 50% of the cells that are going to have the recombinants, right? Why all of a sudden did it change here? It changed because if the genes are very close to one another on the chromosome, there is little chance for recombination. So most of the time, those alleles are going to segregate together. Distance between the genes on the chromosome makes a difference. If they're at opposite ends of the chromosome, one gene's here and the other one's here, you're going to get recombination several times back and forth, many times, and it balances out. They're not linked. The genes are not really linked. But if you get them close together, now one segregation of one allele is going to influence the segregation of another. And guess what? In real life, a lot of diseases occur like this. You have one allele of one gene, and it's commonly co-segregating with a specific allele of another gene, and those two alleles acting together are deleterious to the organism. That's called a haplotype, okay? Haplotype is when those alleles segregate together. Okay, so it's less than half of the time that you would see it. Contrary to this, if they were unlinked, it'd be 50% of the time you get recombinants happening. But if they're linked, and they'll segregate together often. Yeah. This is the last one. Genes A and B are close together on the same chromosome. The genotype of 2 individuals, both of whom are heterozygous for genes A and B, are given below. Is Sveta just as likely to produce gametes A1, B1 as Jackie? Is it critical information that A and B are close together on the same chromosome? Thank you. Okay, so we're looking for this combination. That's a bad one. That means we got trouble. There's prevalence of disease if we have this haplotype. So we're looking for it. Who's more likely to give that into gametes? Are they equally likely, or are they? Okay, so 5, 4, 3, 2, 1. 71% of the class say no, they're not as equally likely to make a gamete that's A1, B1. And who's more likely? Samantha, why? Because he already has those alleles together on the same homolog. So they're likely going to segregate together. Okay, so genes on the same chromosomes do not sort independently, and the chance of recombination depends on the distance between them. They can be so far apart on the same chromosome that they appear to segregate independently, but if they're close together, they often segregate together. Okay, so how we rate the genotype matters. Identify the parental versus the recombinant. Okay, so I want you to practice this. What would be our parental gamete here? Somebody tell me what's the parental gamete for individual number 1. There's 2 possibilities. What's the parental? Yes. A1, B1, or A2, B2. Very good. What would be the recombinants? Yeah.
What was Harvey's 2?
A1, B2, A2, B1. That'd be the recombinants. I'll leave this. You can do this one on your own. How we rate the genotype matters. Parental versus recombinant. So you could, here's your answers right here. Okay, that's it. Kind of rushed again. Remember, you have your evaluation stage due for deep question 1. I'm going to post a little bit of a rubric to give you, there already is the general rubric. I'm going to post a little bit more guidance to help you with assessing your peers' creation. Okay, thanks a lot.